You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Wolfram Zimmermann. He's a professor at the University of Göttingen. We're going to be talking about remuscularization of the failing human heart. And that's one of the interests of his lab, the Zimmermann lab. So, Wolfram, thank you for coming.
Yeah, thank you, Rich, for reaching out. I'm happy to discuss with you our research.
Yeah. So what happens in heart failure? What happens to the heart itself, the muscle, and the other parts? And then what's the solution that you're looking at?
Well, patients with heart failure typically experience a loss of heart muscle cells, which can be the consequence, for example, of a heart attack. It can be a consequence of high blood pressure, infections, and so on. The common problem in all these patients is that they lose cardiomyocytes. They lose heart muscle cells. These are the smallest cellular units which basically provide the contractile force to the heart.
Okay.
And if you lose a significant amount of cardiomyocytes, your heart will weaken, and eventually heart failure develops. And patients experience typically shortness of breath and other symptoms if they have heart failure. And heart failure is, in the end, the most common cause of death worldwide. Because it's a progressive disease, we don't really have a cure for it. Drug therapy, as it is today, at best delays disease progression, but it doesn't really stop it, or it doesn't reverse the disease. And the idea of us and also many other labs is to reintroduce cardiomyocytes into the heart and thereby strengthen the contractile performance of the heart and ideally normalize it.
So what percentage of the heart muscle is impacted in a typical patient that has heart disease? Does it vary tremendously, or is it a certain threshold below which the heart tends to fail?
Well, the defect can vary. We typically estimate that if 25% of the heart is lost, that this then results in severe heart failure. And these would be then the patients that we are targeting with our approaches.
So the cardiomyocytes don't tend to regenerate, or do they only up to a certain point?
No, exactly. Cardiomyocytes are not capable of cellular division, so that means that they cannot regrow. If a certain amount of cardiomyocytes is lost, this then basically leads to a scenario in which the heart cannot compensate the loss in cells anymore, and this is then the time when the heart enlarges and patients experience heart failure. So there's no endogenous repair of the heart.
Well, how do people live to be 80 years old or 100 years old? Their heart is just super old, made of 80-year-old cells, or do they replace themselves?
Exactly. So there's a big debate about this in the field. But it is well known now that there is a very, very limited amount of cardiomyocytes that are being that will regrow during life. So there are estimates that within a normal lifespan, roughly 50% of the cardiomyocytes that you are born with are regrowing. But the rest, the other 50%, it appears that these are the cells that you are born with.
Well, how do people? Okay, so when someone's born, though, they're a baby. I mean, to get to adult status, yeah, they would need, I'm sure, a lot more cardiomyocytes than they're born with. I would think there has to be a lot of replacement going on, at least until someone becomes an adult.
Not really, because the cardiomyocytes are capable of growing in size. So at birth, the cardiomyocytes are still relatively small, and it has been estimated that there is like a 30-fold increase in volume of each cardiomyocyte you're born with.
What does it look like if you compare a baby's cardiomyocyte to an adult's? I don't know if you can do it in humans, but in another animal, what's the morphology differences of the cell? Does it have just more mitochondria, more cytoplasm? Like, how does the cell differ?
Between rodent and human, for example? Or
if you have the example in people, great. But if not, I guess you'd have to go to rodents, between a baby rat and an adult rat, or a baby human and an adult human.
It's mainly size. It's mainly size of the individual cardiomyocyte. It's not really number. So the number is relatively constant over life, but the individual cardiomyocytes can grow in volume. And of course, they are rebuilding proteins all the time. They're rebuilding their contractile machinery all the time. So there's always protein turnover in each of the cell, but the cell itself is not cycling. The cell cycle activity.
So it's the same when the cell enlarges. What enlarges about it? Does it just accumulate more cytoplasm? Does it have more mitochondria? Does it have more? What's different about the cell?
It accumulates the amount of contractile proteins of sarcomeres increases per cell, and also at the same time the amount of mitochondria will increase. That is basically what is happening. So you have more contractile proteins per cell, more mitochondria per cell.
So what happens when someone has an enlarged heart? What's the mechanism there? What are the cells doing?
The cells grow in size. They get bigger. And you have also changes like fibrosis in the heart, but you do not have more cells. So the heart is a terminally differentiated organ for the most part. And small or large heart in kids, adult or patients that are sick differs only by the volume of an individual cardiomyocyte.
So it sounds like the cardiomyocytes are communicating and getting signaling to grow to a certain point, and then they stop, when you're an adult. But I guess in certain dysfunctions, the cardiomyocytes are trying to, do the job they need to do. So maybe they then grow beyond what they should be, and that causes your heart to enlarge. Like what's the mechanism by which your heart enlarges? Why would that happen in an adult?
You described it pretty well. So you have a cardiomyocyte which is constantly under stress. Say if you lose 25% of your heart's myocyte, the other 75% of the cells take over, and to compensate the loss of contractile performance that has occurred, these cells need to grow in size. This is called compensatory hypertrophy, which then over time turns into pathological hypertrophy, and then typically cells start to die. The overloaded cardiomyocytes start to die.
Yeah, why would a cardiomyocyte die just because it got so enlarged? Is it because it's so large the cell can't function and coordinate its activities properly? Can it not beat properly if it's too big, or what's the reason?
Well, there's probably a mismatch of nutrient demand, of ATP demand, and the cell is constantly over exercised. And under these conditions, the cells can go into apoptosis and die. It's not that suddenly 50% of the cardiomyocytes of the heart die. It's a slow process, and over years and sometimes over decades, you have a constant, continuous loss in cardiomyocytes, which then basically results in the clinical symptoms of heart failure.
What do you notice when there's the death of a cardiomyocyte locally? Is it the adjacent cardiomyocytes that go into hypertrophy, or is there a particular pattern of which cells will grow bigger when a given cell dies in an area?
I don't think this is not so clear what happens. If you have a cardiomyocyte that dies, there is typically replacement fibrosis, and the adjacent myocytes then tend to increase in volume. They hypertrophy, and you can have very different pictures of or scenarios. You can have, of course, an infarction with a defined region of the heart being fibrotic, and then you can have patients that did not have a heart attack. They may have other cardiomyopathies or hypertension, and in those patients you can observe a lot of fibrosis throughout the heart. This is called replacement fibrosis, which then leads to issues.
What happens first? Does fibrosis happen first, or does the enlargement of the cells happen first?
This is probably a parallel procedure. If you have cell death, cells die, there is typically replacement fibrosis. But you can also have fibrosis, which can also occur probably without cardiomyocyte death. But these are all mechanisms that are being investigated by many groups around the world, and the exact pathology, the exact changes in the heart are not completely understood. What is clear is that you have a loss in cardiomyocyte, you have fibrosis, and then a loss in function.
Yeah, the reason that I ask is I was imagining if a cell dies and there's fibrosis in that area, that would, I would think, physically restrict the contractile ability of maybe the adjacent cardiomyocytes, because there's fibrosis in the way, it would make it harder for them to contract properly, and maybe they grow larger in response to that. I don't know.
Well, the changes, the heart is the biophysical properties of the myocardium is changing also in heart failure. The heart wall typically gets stiffer. So you're right. I mean, if you have a cardiomyocyte that is located inside a stiff environment, it probably has to contract more strongly. And this may be a process that also causes or contributes to the cell death that we are observing in the failing heart.
And what's causing the fibrosis? Is like the immune system coming in, and literally, where does the fibrotic material come from?
Oh, from fibroblasts. So you have, I mean, the heart is not only composed of cardiomyocytes. Actually, it's for the largest part composed of non-myocytes. And within this non-myocyte population, you have a high amount of fibroblasts, and these fibroblasts are typically there to maintain the extracellular environment, the matrix of the heart. And in a disease scenario, these cells are overactivated or are strongly activated, and then the fibroblasts turn into myofibroblasts and release collagen into the heart, and basically contribute to the fibrosis process.
Okay.
Or they're the main drivers of the fibrotic process, actually.
Yeah. What is your research about specifically? What are you looking to do?
Our research is on. We work on different things in the laboratory, but one of our main research areas is to develop heart muscle using tissue engineering technologies, and to then use this tissue engineered heart muscle as a therapeutic patch in heart failure. So we have been doing that now for many years, and developed basically a number of tissue engineering formats, which we are applying to the heart to strengthen the heart.
You're trying to grow a layer of heart muscle, then implant it into the heart?
Yeah, it's not a layer. It's actually a pretty thick structure. It's like a contractile band-aid, if you wish, which we position directly onto the heart, onto the region of the heart that is dysfunctional.
So what does it look like? Is it a thick patch, or what does it look like?
It's a thick patch, which can be adapted in size and thickness according to the defect, which is of course very different in individual patients, and we tailor our patches according to the patient needs.
So what creature are you growing the structure and then implanting it in? Is it rats or what is it?
Well, we are building human patches, and we have done. I personally started this in 1995, so nearly 25 years now. And of course, at the beginning, we worked in rat, and we worked in mouse, and now for many years we work in human, and have developed heart patches, which we plan to test in patients next year.
So what are some of the challenges you've been running into? Has it worked? Has it failed? What stage are you at?
The challenges are to generate thick patches and to generate them with proper contractile performance. But many of these things have been worked out. And there's clear evidence from animal models that our tissue implants can be used to strengthen the heart, and we have seen in many different species. And one of the main challenges in our program is to develop GMP protocols to basically then have a therapeutic at hand which you can test in a patient.
What do you mean? What does that mean, GMP protocols? Like what are the?
You have to do. In the US, you have to be in compliance with the FDA. You have to produce your therapeutics according to the regulations set by regulatory authorities. And in the US, it's the FDA, and in Germany, it's the Paul-Ehrlich Institute. In Europe, it's the European Medicinal Agency. And so to develop a therapeutic, you have to obey to clear rules as to how to process a therapeutic. And this has been a lot of work over the last, let's say, 4 years at least, to advance our laboratory technology into a stage in which we have a therapeutic at hand.
So what are some of the issues? Have you done this in rats and it works just fine, but in humans you need to go through all this regulation, or what are the stopping points?
Well, to build tissue patches according to regulatory requirements is absolutely not trivial. We have very good evidence that our approach will be pretty safe. Of course, there are some safety concerns which have to be considered. This is, arrhythmias, tumor formation. These are the typical side effects that you could anticipate if you work with stem cell derivatives. And this is what we do. We use induced pluripotent stem cells to derive cardiomyocytes, to derive stroma cells. We mix them in collagen and basically form patches. There's the risk associated with these approaches are unwanted growth of the cells, and another risk is arrhythmia. We have tested, of course, for both in many animal models, and we have so far not observed these complications. So we're pretty confident that our approaches will be safe in clinical applications. We have good efficacy data in animal models, and hope that this can translate to the human. But this will have to be tested in a clinical trial.
Is the tissue beating before you put it into the animal model, or is it only started to beat once it's in the animal model?
No, it's beating before implantation. Constantly, you can keep. We call our model engineered heart muscle, or in short EHM. And EHM can be kept in culture for months, and they will maintain the contractile performance very long.
How do you make sure the two beats come together? Because they probably wouldn't be synchronized.
Beating rate of, that's a good question. So the intrinsic beating rate of our engineered heart muscle is pretty low. It's roughly 30 beats a minute or lower. Human heart is beating faster, and we believe, and we have not. I mean, we have not tested it in human, but we have worked quite a bit in macaque models, and we do have good evidence that the implanted patch is beating in synchrony with the recipient heart. Mechanisms on how that works is not so clear. There can be electromechanical coupling, there can be mechanoelectrical coupling. There are different explanations for this. In rat models, we have seen very nice electrical coupling using high resolution ECG measurements, and yeah, from these data, we are pretty confident that there is coupling or beating in synchrony of the patch with the recipient heart.
Well, how do you couple them? Do you sew the patch on and then shock the heart, or you sew it on and the heart takes over and says, "I'm the boss," and makes the patch fall in line? Or like, how does it work?
Well, we suture the patches to the heart, and then the patch is constantly being trained kind of by the motion of the heart, by the contractile performance of the heart. And we think that this constant tear is leading to a synchronization of the patch, of its endogenous contractile performance. We know that from in vitro experiments, if we use, for example, continuous loading or straining of our tissue, that if we do that at a certain frequency and if we stop then the cyclic loading, that the tissues will continue to beat at that frequency for some time.
So how long does it take for the two, the main heart and the patch to synchronize? And does that depend on the size of the patch that you're putting in?
So we don't know exactly how the time course of integration is. That is also a very difficult experiment to do. Actually, I wouldn't even know how to do it properly. But if you image the heart using ultrasound or MRI, we can see that the patch is contracting basically in synchrony with the recipient heart. We can see that
again, if they're out of whack, that you'd have certain instances when the main heart and the patch both beat at the same time, and then you'd have instances where they beat not at the same time. So I don't know. I would think that would cause the blood flow to go crazy.
Yeah, well, if it would happen, but that we have not observed. I can just, that is in theory, yes, that could happen, but we have not observed any dysfunction or asynchronicity of a heart that has been implanted with the patch so far.
Huh. So you've never seen the main heart go at the rate of the patch? It's always the patch very quickly, synchronizing with the main heart?
That's how it seems to be.
Well, thank God. Good. Okay.
Well, that is where we wonder all the time. But you're right. That this is an obvious question. But we have so far seen that if we expose our tissue patches to mechanical loading, that the tissue patches follow basically the rhythm of the mechanical loading. And we believe that this is happening also in vivo after you implant the patch onto the heart. And the main for us at the moment, we are moving towards clinical applications, towards clinical trials. And for us, the main concern, the main point of our preclinical studies at the moment is to look into safety, is to look into any issues that could occur. And so far, we have not seen any problems after implantation of an EHM onto the heart of rodents or also monkey models.
Oh, so even in monkeys, you you patch their heart and then they have no problem.
Yeah.
It works just fine.
Yeah, we've done that.
Okay, well, that's good. So what's left before you can try it in people? What do you have to do?
We're presently filing a clinical trial application, and we have pretty much finished our preclinical program, and we anticipate to have the first patient treated in the first half of next year.
Have you noticed that so are you cutting out the part of the heart that's damaged and then suturing in this new patch, or are you just suturing over the damaged part?
We're suturing over the damaged part. The ideal scenario could be to cut out the damaged part and then basically replace that with a tissue patch. We're working on that also experimentally, but this is, at least in my point of view, not something that you can that we can try in patients. So it is. We are the main point is to add cardiomyocytes to the heart. We're pretty confident that we can do that, and to do that at low risk for the patient. And we're also pretty confident we can do that. So we will probably have to select patients very carefully and not treat patients with a very stiff scar. So that could be an exclusion criterion for our approach.
Where are you getting the cells from? From the patient themselves and culturing them?
No.
Or where do they come from?
We get them from induced pluripotent stem cells, but the patches are not autologous. They are an allogeneic. This is an allogeneic approach, and we have basically we have a GMP IPS9 in the process to develop sufficient amount of cardiomyocytes and stroma cells and tissue patches for clinical applications from that. But the patients will have to be immune suppressed to not reject the patch.
Well, why not try to culture them from the person themselves?
That's something that has been proposed ever since the IPS technology came around, but that's pretty unrealistic. First of all, it takes approximately 2 years to have a tissue patch done from an individual patient. That is way too long for too long of a preparation time. And then even if you implant autologous material, at least in our experience, there are immune responses that even cause rejection or destruction of an implant. We've seen that in our monkey model, in which we have done also autologous implants. And for two reasons, it doesn't seem to be a logical application. First of all, for logistic and cost reasons, and also the cell biology is not so simple. So if you process cells,
why do you think that? Why do you think that the tissue is getting rejected if it's I guess you say autologous?
Well, there's a lot of if you have cells in culture for extended period of time. There is autoantigen expression, cells acquire mutations in mitochondria. There's a lot of what is that? Well, frankly, we don't know exactly, but there are a couple of published observations which support that even an autograft would be rejected.
Huh. Oh, that doesn't really make sense, but
no, it doesn't. I mean. Well, it seems so easy just to take cells from a patient and then grow something, that can be done, but it's an unrealistic option, I would say.
Have you tried to take a couple of cells from a monkey, from their heart, and put them in the dish with the patch and see if there's crosstalk or if it changes the behavior of the cells of the patch?
We have done. We have generated implants from the monkey's own cells, and then we put these implants back into the very same monkey. So that we have done, and what we have observed is rejection.
Is the rejection the same as if you're using, non-autologous tissue, or is it a different type of rejection? faster, slower?
The type of rejection, well, that could be different. We would anticipate that if you implant a patch from a different monkey, an allograft, that there is strong rejection. That we have not tested. We have always administered immune suppression. At least the monkeys, you cannot, wouldn't make any sense to do these experiments. You would lose the graft.
So
the experiment that made sense was to do an autograft, and of course we anticipate that the grafts would survive, but that's not always the case.
So what happened? How long did it take? Did the whole thing just become fibrotic, or like what happened when you did the autograft?
Well, we have done 2 monkeys with autografts, and we have studied them for 3 and for 6 months. Keep in mind, these experiments for each of these monkeys takes 2 years to do. And in the monkey that we investigated 3 months after implantation, we did see still cardiomyocytes, but we also saw inflammatory responses. And in the monkey that we investigated 6 months after implantation, not a single cardiomyocyte could be observed.
So after 6 months, the whole patch was it was fibrotic, or what happened to it?
It was basically a cell-free. Well, it was a cell-free patch basically afterwards. Wouldn't really call it fibrotic. It's not. We didn't see really an active, thicker fibrotic area. It basically looked like a cell-free patch.
What do you mean when you say it's cell-free? Did the cells leave?
They died.
They just died.
Well, if they die, if the body clears dead cells. So if there is an inflammation, the cells, you have leukocyte infiltration, macrophage infiltration, and you have then basically clearance of the dead cells. This is also happening.
So you just had what, extracellular matrix and full of dead cells, or full of just gook? There's no living cells there.
Well, after 6 months, there is no cardiomyocyte left. In this autograft animal without immune suppression. With immune suppression and allografts, we do have a high retention of cardiomyocytes after 6 months. So we were kind of testing whether or not in an autograft without immune suppression, we thought that there could be cell retention, and for 3 months in one macaque there was, in the other one, which we studied for 6 months, there was no cardiomyocytes left.
So what? It just sounds like a tough trade-off. If I'm older and my heart is damaged and I need this graft, I can get it, but then I have to be immune suppressed. So how long will this work until I don't know, I succumb, or it stops working, or works forever as long as you're immune suppressed?
Well, this is like in a normal heart transplant. If you have a heart transplant, you are under immune suppression until the end of your life, and of course there are side effects. There can be side effects, but these are you have to watch out for that. But it's
too
not
bad
such
that you have that gigantic trade-off. I just wonder if there's
it's not so gigantic. If these patients that we're treating are dying, we're talking about an end-stage disease scenario in which patients have a life expectancy under normal conditions for 12 months. Yeah, and that's
okay.
You will not have tumor formation or other complications really.
So what's the life expectancy with the patch in place and the immunosuppression?
Well,
is there a ballpark idea? Well, what's your guess? Can you predict it at all or no?
Well, we can predict that the patch will enhance contractile performance of the heart. And of course, the anticipation is that patients would live longer with a heart patch. And finally, of course, the plan is to really replace the scar with contractile muscle. But making any predictions now as to how much longer would a patient live, this is something that you have to test in the clinical trial.
Well, maybe you could get some information by looking at heart transplants. How long do people tend to survive after a heart transplant, and what is it that gives out and kills them?
Well, after a heart transplant, you have a pretty normal life expectancy.
Oh, okay, I thought it was diminished. I didn't know that. I
Not
thought
well, not much. Not well. Survival rate in heart transplant patients is pretty good, pretty encouraging, because if we had enough heart transplants, we would not need any tissue patches. This is kind of the problem we're having. If you had a heart transplant for everybody, then you would not need any cells to be implanted into a heart. But we will never have enough hearts to be transplanted. So we have to find solutions for the patients that are today dying on a transplant waiting list.
So what are the mysteries involved in this process to you? What either surprises you or really piques your curiosity about it?
Well, it's amazing to see that immature or stem cell derived cardiomyocytes have the capacity to self-organize under appropriate stimuli into a pretty mature heart muscle. And it is also then surprising that these tissue patches, after implantation, in the absence of vascularization, at least at the time point of implantation, there is no vascularization of the graft, that these cardiomyocytes survive, start to mature further in vivo, and then also support the heart. We find this pretty amazing and also surprising, of course.
What's directing the cells, do you believe, to form in a certain way, to beat in a certain way, to organize? Like where do the plans of organization and all that, where are they held, do you believe, and how are they communicated amongst the cells?
We think it is primarily mechanical impulses. The stretching of the patch after implantation, also in our in vitro preparation of tissue patches, we use mechanical stimulation to train the tissues, and that is the main principle of our tissue engineering concept, that we always use mechanical triggers to train our tissue, and then after implantation, the heart takes care of that.
Well, I thought that the heart was primarily electrical, and it used electrical signaling as well. So do you see evidence of electrical signaling or just mechanical signaling in the patch? I mean, have you tried to look at the cell-to-cell communication? Is there chemical gradients? Are there electrical gradients being set up? I mean,
Well, you can study this very easily and nicely in the laboratory. I mean, there are clear electrical circuits that are forming, and there's a pretty well developed functional syncytium in the laboratory already. And then after implantation, there's good evidence that these patches then also are synchronized with a normal heart, with a recipient heart.
Right.
That is certainly not by direct electrical coupling direct after implantation, because we basically implant a patch onto the heart, and this is not a plug-and-play system. Maybe the mechanical impulses from the recipient heart lead to activation of contractility in the patch. This is something that is at least what we hypothesize.
What about the patch itself, the edges of it versus the center? Are the cells different? Or the top versus the bottom? Are they pretty uniform across the whole patch?
They're pretty uniform. They're pretty uniform, which is also surprising, because of course you would think that if you generate something of significant thickness, that you would have a preferential growth of cells at the outside of patches where there may be more oxygen. And that may indeed be true for very thick patches, but in the patches that we have generated so far, we have not observed a preference for outside versus inside. That may be because we are designing patches in a way that they are that they receive nutrients also inside the tissue.
What about the size of the cells in the patch? Is there any correlation between the thickness or size or dimensions of the patch and the size of the cardiomyocytes in them? Do they sometimes get bigger and you don't know why, or do they stay a certain size? Or
the cardiomyocytes inside the patches, they are stem cell derived cardiomyocytes. They are less mature and smaller as compared to the cardiomyocytes in the adult heart, but they have the size of postnatal cardiomyocytes. So they resemble basically a young heart. which should still be capable of growing in size. We expect that there is hypertrophy after proper, if the proper environmental stimuli are present.
But that may be an issue. If you sew a patch on and the cells are small, and then they start getting bigger once they're implanted next to the heart, wouldn't that strain the suture and change the morphology of the patch?
No, not really. Well, no, not really. Actually, this is a wanted effect, because we want to have the reason for cardiomyocyte survival in our case is certainly that these cells are metabolically less demanding as compared to a big adult cardiomyocyte. This is well known that if you take a cardiomyocyte from an adult cardiomyocyte from the heart, if you would, these cells are they need a lot of oxygen, and they would die if injected into the heart or implanted onto the heart. So we intentionally keep the cells.
If they grew bigger again, they would push other stuff out of the way. They would pull on the suture. They would get bigger.
No, not really. No, I don't think so. Well, we have not observed that. They're trained by this we have never observed.
How much smaller are they than the adult cardiomyocytes? On a cell-to-cell basis, a little bit smaller, a lot smaller.
They are roughly tenfold smaller in volume.
Wow, that's a huge change. I would think that would be. That's crazy.
Yeah, they have the size of they have the same length of adult cardiomyocytes, but they're thinner.
Oh, interesting. Have you implanted patches and then gone in there after a period of time and looked at the heart and pulled the patches out and see how they changed?
We have not. We have done, of course, histological assessments, and we see that the cardiomyocytes are growing in size. It's pretty difficult to quantify, to precisely quantify cardiomyocyte volume in a tissue section. It is much easier to do that after isolation of cells from a tissue patch. And here we have done these volume comparisons, the cell volume comparisons.
Well, what if you made a patch in the shape of like a circle or the letter C, and then you patched it on in a rodent heart, and then 6 months later you euthanize the animal, looked at the heart, and see if the shape of the patch changed. If it had a very specific shape going in, it might be easy to observe it change. Maybe you make a shape like a star or a C or something.
No, you can still see of course, the patch you can still see after months. That's the patch is still present. Taking it off the heart is not so easy. You would have to basically scrape it off the heart, and thereby you will probably destroy the patch pretty much. There is a clear or a strong connection of the tissue patch with the recipient heart.
Okay, and then just a couple more questions. Hope I'm not driving you crazy with the questions, but I don't know if you considered these before, so maybe at least it's new stuff, maybe not. But what was I going to ask you now? So,
go ahead.
you said the patch. What is the suture going into? So is the suture just in between the individual cardiomyocytes on the patch side, and then in between the cells on the other side, or is the suture what are you suturing into? Is there a border of the patch?
The patches have roughly 4 by 4 centimeters in XY dimensions, and that can be extended or decreased. That's you can do it pretty much in any size, and then you basically use a suture and you tie. You stitch through the patch and through the heart, and you tie a knot.
But what does the suture bite into? It just goes in between the cells and extracellular matrix.
It goes through the patch. Yeah. Well, yeah, you can see the cells obviously. So you just go directly through the patch and directly into the heart, and then you tie it.
So have you seen any pattern of fibrosis that happens over time? If you observe the patch that's implanted after 6 months or a year, does the area around the suture tend to die, or the whole thing's preserved? Like,
Well,
what does it look like?
there's always, if you put a foreign material into the heart, like a suture, there is some foreign body reaction to the suture, and you can see the suture easily after 6 months. This is the longest time we have studied patch survival in a monkey model. So depending a bit on the suture material, there are different suture materials. Some materials resolve over time, others don't. And in the regions of the stitch, you will see some very small fibrotic changes.
Okay. Well, very good. What's your ideal? What do you want to have happen in the next couple of years? What would be a great goal for you?
Well, we hope that the approach we are developing can be safely applied in patients, and will then help to support patient hearts or patients with heart failure. And we are, of course, super excited to see the results of the clinical trial we are starting next year.
How long do you think this will take before real human patients can use it?
Before it is, well, that depends on the outcome, of course, of our study. We're planning on treating.
Well, if everything goes well. If everything goes well, how long do you think it'll be?
Well, if it goes well, we know in 3 years we know whether or not it goes well, and then it depends on how well it went. We will have to see. That this is, you can imagine, these are very difficult patients to treat. They have very limited options. And one thing we know for sure is that we can add cardiomyocytes to a failing heart. So that's very clear. And we also know that we can maintain them for a long period of time. So there are several questions that we cannot answer. Whether do we need more cells? Do we need them integrated in a different way? Do we need to combine tissue engineering approaches and cell injection approaches? Are there specific patients that will benefit from cell injections and not from patches, and vice versa? So these are kind of open questions which I think the field is ready now to test this in a clinical trial.
Okay. So what's the best way for people to get in touch, with the lab and see what you're doing, and
well, they can just they can just Google for tissue engineering and Göttingen and my lab, basically. So they can easily come in touch with us.
Okay. Well, very good. Well, Wolfram, thanks for coming on the podcast. I appreciate it.
Yeah, sure, Richard. And I hope this was useful.
Oh yeah, no, definitely. You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.